Viruses, Part 1. Ooh. Viruses are tiny infections, a cellular agent having either DNA or RNA. Viruses does not have a cell, but viruses have a genome. A viron is a virus outside of a cell, consisting of a proteinaceous capsid surrounding a nucleic acid core. Prions are proteinaceous infections particles that lacks nucleic acid and replicates by converting similar normal proteins into new prions. Prions mutation cause proteins to have different folding properties. A viroid is a naked RNA molecule that infects plants. How do viruses differ from living organisms? Viruses are not living organisms because they are incapable of carrying out all of life processes. Viruses are not made of cells, they are acellular. Viruses cannot reproduce on their own. Viruses do not grow or undergo division. Viruses do not transform energy. Viruses lack machinery for protein synthesis. Characteristics of a virus. What are viruses made of? Viruses have a nucleic acid, proteins, and sometimes lipids. The nucleic acid in a virus is surrounded by a protective protein coat called a capsid. A nucleic acid plus a capsid is called a neocapsid. An outer membranous layer called an envelope made of phospholipid and proteins surrounds the capsid in some viruses. Naked viruses lack envelopes. The capsid morphology. The protein coat called the capsid provides a protection for viral nucleic acid and a means of attachment to a host cell. Capsids are composed of proteinaceous subunits called capsamers. Some capsids compose a single type of capsamer, others composed of multiple types. The viral envelope. The viral envelope is acquired from host cell during the viral replication or release. The viral envelope is composed from the membrane system of a host. The viral capsid is composed of phospholipids and proteins. Some proteins are virally coded glycoproteins. The glycoproteins are known as spikes. The envelope's proteins and glycoproteins often play a role in host recognition. Now I got a question for you guys. How are viruses classified? Viruses are a type of nucleic acid, either containing DNA or RNA. See, the key word here is or. They never contain both. DNA viruses contain DNA as their genetic material, and viruses contain RNA as their genetic material. Okay, now we're going to talk about the viral structure. The viral structure is a viron, and a viron is a virus outside of a host cell. Not outside of a host cell. A viron is a virus outside of a cell consisting of a proteinaceous capsid surrounding a nucleic acid core. And the virion is a fully developed particle. The virion consists of a nucleic acid, a capsid, an envelope, and spikes. The nucleic acid is either DNA or RNA, and this nucleic acid can be single-stranded or double-stranded. This nucleic acid also can be linear or circular. The capsid within a virion is a protein coat made of capsomers. The capsomers are the subunits, guys. Remember that. The envelope within the virion is a lipid protein and a carbohydrate coating on the same viruses. The spikes within the virion are projections from the outer surface. Okay, now we're going to talk about the shape of a capsid. A capsid can be helical, polyhedral, or complex. Helical consists of capsomers bonded, and a helical is a spiral shape. A polyhedral have flat sides, and a complex capsid have many different shapes. Helical, polyhedral, complex. Okay, the helical virus is hollow and cylinder-like capsid. The polyhedral viruses have many sides. The enveloped viruses. And then we have the complex viruses, which are complicated structures. The orthopox virus. Smallpox is an acute contagious disease caused by variola viruses, a member of the orthopox family. It was one of the world's most devastating diseases known to humanity and was declared as eradicated in 1980, following a global immunization campaign led by the World Health Organization, known as WHO. Kinds of hosts that the orthopox infect: plant viruses, bacteriophage, and animal viruses. Plant viruses infect plant cells, bacteriophage infect bacteria cells, animal viruses infect animal cells. Viruses sizes. The bacteriophage F2 and S2 is usually 24 nm. The polioviruses and the rhinoviruses usually are the same sizes. These viruses are 30 nm. The adenoviruses are usually 90 nm. The rabies viruses are usually 170 times 70 nm. The prion viruses is usually 200 times 20 nm. The viroid viruses is usually 300 times 10 nm. Okay, now we're going to move on to the replication of animal viruses. These viruses reproduce via 3 basic steps. The first step, this virus delivers their own genomes into a host cell, known as attachment and entry. The second step of replication of animal viruses, these viruses command the host cell transcription and translation machineries and utilize host cell building blocks to copy viral genomes and synthesize viral proteins. This process is called biosynthesis. The third step of replication of animal viruses is the viral genomes and proteins are self-assembled and exit the host cell as no infectious particles. And this process is called assembly and exit. Attachment of viruses to the host cell. How do a virus recognize a host cell, you may ask? Specifically due to affinity of a viral surface proteins or glycoproteins. And remember, the glycoproteins are known as spikes. on the surface of a host cell? A. Capsomers on viral capsid for non-enveloped viruses. B. Proteins or glycoproteins on enveloped for enveloped viruses. Okay, now here's the second answer to how does a virus recognize a host cell. The recognition may be so specific that the virus may only infect a particular cell type of the host organism. For example, HIV only attacks helper T lymphocytes, a type of white blood cells in humans. Lymphocytes, a type of white blood cells in humans. Entry of viruses into a host cell. Not completely understood, but appears to be three methods. 3 ways a virus enters animal cells. 1. Direct penetration of naked viruses. 2. Membrane fusion. 3. Endocytosis. 1. Direct penetration of a naked virus. 2. Membrane fusion. Membrane fusion consists of envelope attaches to the host cell. Envelope becomes a part of the host cell membrane, and the neocapsid, which is the nucleic acid in a capsid, enters a genome and it is unencoded. 3. Endocytosis. Have 4 steps. The envelope attached to the host cell. The envelope virus is endocytoted. And the endocytoted virus enters the cell, and the neocapsid enters and a genome is unencoded. Okay, now we have the synthesis of a viral nucleic acid and proteins. In order to make a new virus particles, we need to make copies of the genome, which is the DNA or the RNA. In viral proteins, capsomers or spikes or glycoproteins. Basic biology review to help understand the viral replication of biosynthesis. A. In an animal cell, DNA in the nucleus is transcribed into mRNA. B. The mRNA moves out the cytoplasma. And then C. The code in the mRNA is translated to form a protein molecule. So we have DNA, which is transcribed into mRNA, and mRNA is translated into a protein. Synthesis of a viral nucleic acid and proteins. Depends on what type of nucleic acid, DNA or RNA, double-stranded or single-stranded. So first, the first synthesis step is how is RNA synthesized? Meaning, how is RNA used as a template for viral protein? The second question we have here is what molecules serve as a template for nucleic acid replication? The pox virus is a double-strand DNA virus that encodes its own transcriptase and does not need to enter nucleus of the host. The pox virus is complex and enveloped. The rhinovirus is polyhedral and non-enveloped. The rabies virus is helical and enveloped. The influenza, known as the flu virus, is helical and enveloped. RNA viruses that copy their RNA to DNA are called the retroviruses. Assembly and release. Naked viruses, after construction of capsid, released from the animal cell through 1. Exocytosis, or 2. May cause lysis and death to cells. Enveloped viruses, often released through a process called budding. Acquire envelope as emerge from plasma membrane of host cell. Note that some viruses acquire envelope from nuclear membrane or that the endoplasmic reticulum of Golgi of the host cell. Golgi of the host cell. Hidden viruses. A. Latency occurs when animal viruses remain dormant in host cells. May be prolonged for years with no viral activity, signs, or symptoms. Some latent viruses become incorporated into the host chromosome. When viral DNA is incorporated into the host DNA, condition is permanent. And then it becomes permanent physical part of the host chromosomes. Examples such as HIV, chickenpox virus, and herpes virus. The herpes simplex virus life cycle. Herpes simplex virus, HSV, is shown undergoing the lattice cycle in epithelial cells of the skin to cause a primary infection. Some viruses enters the sensory neuron terminals and travels retrogradely to the nucleus where it establishes latency. Periodically results in integrate transport of viral particles shedding from the neuron and reinfections of epithelial cells, which leads to a symptomatic shedding of recurrent lesions. Viral diseases of the skin. So we have the measles, which is a causative virus. The measles virus, also known as the morbillivirus, is gram-positive, single-stranded RNA. This virus is enveloped and has a helical capsid. Virulence factors of the measles virus. We have hemagglutinin and fusion protein. The hemagglutinin attaches to receptors on the host epithelial cells. The fusion protein has the ability to fuse host cells and cause synthesia, giant cells with many nuclei to form. The clinical features of the measles virus last 3 to 4 days, and then you may begin to notice a sore throat, a dry cough, a headache, or conjunctivitis, swollen nose, and fever. 9 to 11 days in child and 21 days in an adult. Characteristic oral lesions called the Koplik spots. Symptoms by 2 to 3 days. Rash erupts on head and spreads to trunk and extremities until most of the body is covered. Spreads over 3 to 4 days. Rash forms red patches and gradually fade to brown. This is called maculopapular. Transmission of measles include droplet. Viruses enter through the nose, mouth, and conjunctive. The conjunctive is the eye area. Diagnosis of measles. You only can diagnose measles if you notice signs of measles. Prevention of measles include a live, weakened MMR vaccine. Distinguishing features. The rash is maculopapular, which means the rash is flat to slightly raised or colored bumps. This often leads to secondary bacterial and ear and sinus infections. Epidemiology of measles. Measles is highly contagious, spread via respiratory droplets. Humans are the only host. Vaccinations has reduced the spread of the measles disease. Herpes infections. Herpes comes from the Greek word to creep. Herpes is a causative virus known as human herpesvirus 1 and 2, HHV1 and HHV2, originally called the herpes simplex, HSV1 and HSV2. The herpes virus is double-stranded DNA. The herpes virus is enveloped, and the herpes virus is has a icosheteral capsid. The virulence factors of a herpes virus. The herpes virus have an enveloped glycoprotein. The herpes virus have adhesion to the host epithelial cells. The herpes virus has fusion of host cells to form syncytia, which means move from one host cell to the next to avoid antibodies. The herpes virus is an inactivation of complement. After initial infection, constantly begin produced at a low level in some surface cells, causing continuously shedding of virus even in the absence of the lesions. Clinical features of the herpes virus. The lesion sites. So you can notice herpes on the lips, on the fingers, on the conjunctiva, which means the ocular level. Ocular is known as the eyes, and the genital areas. So the herpes on the lips are also called cold sores. Cold sores, you might notice sores around the lips. The fingers, you might notice swelling around the nail bed or sores around the nail bed. The conjunctiva is ocular herpes, so anything within the eyes, such as swelling, dry eyes, red eyes, crusted eyes. The genital herpes, you'll notice sores on the genital areas. The lesions on the finger site of herpes is called the Whitlow. Ocular herpes are within the eyes. Oral herpes lesions are around the mouth or in the mouth. Pathogenesis. Initial infection. The virus replicates in epithelial cells near the site of infection. This virus leads to inflammation in cell death. Inflammation and cell death. You might notice blisters begin to form and fill with amber fluid that contains many major virions. Once again, virions is a virus outside of a cell consisting of proteinaceous capsid surrounding a nucleic acid core. The latent infection is a virus that remain inactive in nerve cells in the body. With the latent infection, you may begin to notice pain, tingling, burning, or itching may signal an outbreak of the latent infection. Outbreaks tend to decrease in frequency over time. Transmission of the herpes virus. You only can get herpes through direct contact, and you can diagnose herpes from the presence of the characteristic of the lesions, such as the lips, fingers, and the conjunctiva is one more. The fingers, the lips, the conjunctiva, and the genitals. The treatment for herpes is a chemotherapeutic. Chemotherapeutic drugs help limit the duration of the outbreak, but do not get rid of the outbreak itself. Prevention of herpes. Healthcare workers wear gloves. Epidemiology of herpes. So for the HHV1, the initial infection is usually asymptomatic and is acquired by 80% of children worldwide by the age of 2. HHV2 is acquired between ages 15 and 29. And in this age group, 15 to 29 is usually sexually active. Both can be spread between mucous membranes of the mouth and genitals. The herpes whitlow, which is herpes around the fingers. People usually get these herpes due to thumb sucking. Those with genital herpes that decide to touch their genital areas, and now the disease or the infection is now on the fingers. And dentist/dental hygienist. Herpes infection in adults are not life-threatening. Neonatal infections can be fatal, meaning that babies with a herpes infection can be in some trouble. You have to stop.